if I can just reintroduce him perhaps and say that he has been absolutely instrumental in informing what we are trying to achieve here with prostate cancer detection. And one of the things that, let me pin myself here so you can see me and I can see myself. There we go. Now, what it all started with is this. So we have here a poster that we presented at the Prostate Cancer Foundation annual meeting, and it had to do with GCMS, volatile organic compound analytics. And to be very, very clear, there's two very different ways to sniff out disease. One has to do with analysis, which is not what the dogs do, categorically so. Analysis means you're telling me what something's made of. That is not the same as telling me what something smells of. They are related. The volatile organic compounds that are coming off of everybody's body and that communicate their body scent, and they can communicate whether someone is fighting a disease, if their immune system is responding to something like COVID or cancer. The analogy that is correct is to think of the disease leaving a footprint in the sand and the volatile organic compounds being the sand particles. Grains of sand are not the footprints. Okay. Of course, being able to see the grains, being able to see the outline of a footprint is important. And that is what happens when you train these dogs. And we trained several of them to Claire Guest's outfit in Milton Keynes, UK, called medical detection dogs, on the most powerful, scientifically rigorous and impactful charities on the planet, right alongside there with Prostate Cancer Foundation, another scientifically rigorous and impactful charity. One accepts a lot of donations and gives out a lot of money. Medical detection dogs accepts donations and creates fantastic solutions to people. So what is important to understand is that when you do GCMS, which is this kind of diagram that you end up getting a list of molecules by name and concentration, is very different than what the dog is doing. However, they are related because in both cases, the signal rides on the volatiles. So we detect the volatiles themselves in a GCMS setting, and then we let loose Stephen Thaler's AI to basically emulate what the dog's brain is doing. So we have artificial dog intelligence now, and we have shown with the startup that Bruce is an advisor to, and I'm so honored to be his co-author and the co-founder of this startup that is aiming to identify whether someone is suffering from prostate cancer, and perhaps even be very ambitious and try and do what's called binning by Gleason score. That means not only tell you whether you have this disease, but also tell you how aggressive it might be, which is the key to informing the treatment. If you overtreat this disease, you might lose function, you might lose quality of life, or you might even lose your life as a result of treatment. To be able to avoid unnecessary treatment is one of the greatest challenges and opportunities that this technology is capable of delivering to us. To be able to say, look, you have this type of cancer, let's treat you this way, is just so paramount. So we presented this work, and I am so thankful to Bruce, who is about to come on and tell us his perspective on evolution and cancer. So I will stop sharing my screen and welcome Bruce again, who will be assisted by Matt in presenting his slides. So, and I see that Dr. Adam Feldman has joined us too, and he is the next to speak after Bruce in 5 minutes. Bruce, take it away.
Okay, Matt, can I have my slides? Okay, great. Next slide, please. Or can I control them? So cancer isn't new. A lot of times people think cancer is something that's relatively new. But here's an example, the oldest one that we think has been identified in an early human ancestor. So almost 2 million years ago, this was a tumor in the, in the metatarsal that was found in South Africa. Next slide, please. So what I'm going to talk about a little bit, the study that Thomas mentioned, where we're planning on expanding upon the initial study, one of the things that I'd like to do is, in parallel with looking at the VOCs the way Thomas and Andreas described, I'd also like to look at the metabolomics. And I'm sure most of you know this, but metabolism is basically the chemical reactions that support essentially our life. And what's interesting about metabolomics, it's sort of the final readout of genomics, transcriptomics, proteomics, and metabolomics is really the effectors that are producing the phenotype. Next slide, please. So one of the hallmarks of cancer is that metabolism gets dramatically rewired to accommodate a huge increase in cellular proliferation. So at the top, we see a normal resting cell, and it basically uses oxidative phosphorylation to take glucose and make 36 moles of ATP. Mostly the big function is for energy. But cancer cells that want to proliferate a lot use a much less energy efficient process of aerobic glycolysis, where they only produce 2 moles of ATP, but they produce lactate, and eventually they produce macromolecules, nucleotides, amino acids, lipids, that can be used to form new cells, which is what cancer wants to do, and also to control the oxidative stress that comes in that process. Next slide, please. So here's an example. These are VOCs that have been found most frequently associated with cancer. So if you look at the top there, you see a triangle that has an 8 on it. And that corresponds to hexanal, which has been found to be associated in at least 8 different cancers, and so on down the diagram. And most of the VOCs that have been associated with cancer have been in these 5 categories here on the right: aldehydes, ketones, hydrocarbons, aromatics, and alcohol. Next slide, please. So how did these metabolic pathways evolve? So we know that this process took place over billions of years. Amino acids are probably the oldest of the essential molecules that came about 3.5 billion years ago. And over this time, the biggest change was that originally primitive organisms were living in this sort of primordial soup that provided all the organics that they needed. And eventually, as essentially the population grew, some of these organics started to diminish. So what evolved was the ability of biosynthesis to synthesize the necessary compounds. But this took place over billions, maybe millions of years. Next slide, please. So let's think about human history. And this is why I talk about we've been outrunning evolution. Next slide, please. So about 1 million years ago, we acquired the ability to use fire. And through most of human history, getting food was difficult. We often didn't have it. It required a lot of energy. And sometimes it required diverting energy that you needed just to keep warm. And another huge change is back 1 million years ago, there was a tremendous variety of plants and animals that could be used for food. And this has dramatically decreased over time. Next slide, please. And what we have seen only in about the last 100 years is that food is readily available for most of the world, more than we need. We don't have to work hard to get it. We don't have to spend a lot of our energy keeping warm. And as a result, we've seen this huge increase in obesity. Next slide. So if you look at the US in 1992, the brown states had fewer than 50% of people overweight, and the purple ones, 12 of them, had 50 to 55%. Next slide. 5 years later, I think it was, more than half the country had more than 50% overweight, and a few states had more than 55%. Next slide. So 6 years later, almost the entire country was at least 50% overweight, and about half was 55% overweight or more. Next slide. 15 years later, the entire country had more than 55% of people overweight. That's pretty amazing that they accomplished that. We accomplished that in 15 years. And if you think about it, cigarettes are one of the probably most powerful lifestyle carcinogens. It took 20 years from the increase in smoking to start being reflected in increase in cancer. So we haven't even seen what's going to happen with obesity, nor have we seen what's going to happen when not only are people obese, but in utero, people are exposed to the hormonal milieu of an obese mother. Next slide. Okay, so how does that relate to prostate cancer? So I'm interested in the metabolomics that we can measure in prostate fluid. So the prostate produces fluid, which is combined with fluid from the seminal vesicles to form semen. And when we get a tumor removed, when we get a prostate removed, it goes to the pathology lab, and the pathologist literally squeeze the prostate like a sponge, and you can get some of this prostate fluid to come out. And this is from the environment where the cancer is formed, the ducts inside the prostate. So we think that it might be highly specific to what's going on that could be causing the cancer. Next slide. So what we did, we identified a number of Black men who'd been treated for prostate cancer. Some of them were apparently cured, the tumor never came back, and some of them had the tumor come back. And we matched them on age and a whole bunch of other factors. Then we did the same thing for a group of white patients. And we looked at their prostate fluid and we measured metabolites in them. And so here's a random forest plot of the top 30 metabolites in each of them. And if you look at the Black patients, you can see a preponderance of the orange circles, the amino acids, whereas in the whites, you see a preponderance of the sort of turquoise circles for lipids. So different patterns already. Next slide. So then using some more bioinformatic approaches, we came up with, and of course this is very preliminary, it's a small sample, and we've actually received funding from the DoD to expand this study. But we found 3 metabolites in Black men and 4 completely different metabolites in white men that were all associated with about a doubling or more of risk of having the tumor come back. So we're hoping to continue doing this kind of research in the study that Thomas mentioned, where we can compare what we're finding in the VOCs from urine with what we find in the prostatic fluid. I think that's it. Next slide. Next slide, please. Yep. Thank you. I hope I stayed on time.
That was fantastic and so powerful. And I am impressed and surprised by these data, and so very interesting. Wow, is what I have to say. And for our next speaker, we have Adam Feldman, Dr. Adam Feldman, who is a urologist and oncologist and the director of the Harvard Urological Oncology Fellowship at the Massachusetts General Hospital Department of Urology, and he's also an assistant professor of surgery at Harvard Medical School. I'm also very honored and happy to have him as an advisor and co-founder of Sentient, which is our new attempt to mimic the dogs with artificial intelligence and use GCMS to predict even perhaps what the Gleason score of someone might be. Adam, you have 5 minutes. Take it away.
Great. Thank you. Thank you, Andreas, for welcoming me. I will apologize up front if my eyes are sagging a little bit. I'm on call this weekend and was in the middle of the night taking care of a patient with a kidney stone. So I'm going to share my screen. Great. And hopefully everybody can see that. Great. So, so I just want to talk a little bit of the practical aspect of, why early detection is important. I'll focus on prostate cancer, but really it's applicable to many malignancies that, we want to identify patients who have cancer that needs to be treated. And I'll clarify that comment. We want to identify them early before there's a greater risk of metastasis and spread. And that's the whole goal for screening and biomarker development, and really stresses the importance of that. So prostate cancer, just to focus on that for this part of the discussion, prostate cancer, it's the most common cancer in men. In 2020, there were over 190,000 new cases. It's the second most common cause of cancer death, and then and over 33,000 cases in 2020. And about 1 in 9 men will develop prostate cancer in their lifetime. And just to talk about screening for prostate cancer and incidence of prostate cancer, this is just a very good, this is an image that shows incidence of different malignancies, and the brown line is prostate cancer. And you can see that in the early 90s, the incidence shot up. And the reason it shot up is because that's when we started using a blood test called PSA, or prostate specific antigen, to screen men for prostate cancer. And that led to an increased incidence just by finding them. And then you basically, as they were found and the incidence sort of settled out at a higher level than previously, hopefully you can see my arrow. And then an interesting thing happened then here later in, sort of after around 2010 or so, what's called the U.S. Preventive Services Task Force is an agency that looks at screening tests. There was controversy as well about mammography that they had addressed, but they had recommended that PSA not be used for screening for prostate cancer. And what you can see is that the incidence of prostate cancer at this point actually declined. And I'll get to what the sort of implications of that decision were. Here you can see that when after PSA testing began, that the actually mortality from prostate cancer had actually decreased over time. Again, I'd mentioned in 2010, 2011, it was actually 2011, the panel, the U.S. Preventive Services Task Force, had said that PSA testing should not be used for prostate cancer screening, and that was based on 2 randomized controlled trials, which I would argue that people who take care of men with prostate cancer probably interpreted the results a little bit differently, and there was some difference in interpretation. And what we had seen since that time is that after that decision and after PSA, and the result of that decision is that in the primary care clinics and sort of internists' offices, the rate at which men were being screened significantly went down. And what we found, and what it was sort of what we had worried would happen, is that we ended up seeing a decrease in men presenting with localized disease, but an increase in men who present with sort of regionally advanced disease and distant disease. So, really this unfortunate, finding confirmed that early detection really is very, very important and critical to helping prevent progression from prostate cancer. So with that information and in the years since then, in the past few years, the agency backtracks. They said now, okay, PSA testing should be an individualized decision. We know PSA, which is again the most commonly used biomarker for screening for prostate cancer and detecting prostate cancer, it's not perfect. It's far from perfect. We know that. So are there other markers that have been developed? There are many markers. There's markers that are blood-based or serum-based, urine-based, tissue-based, to try to improve our screening for prostate cancer and our ability to identify men who need treatment. And the reason I say men who need treatment is we know that there are many cases of prostate cancer which may not need to be treated. Many patients with what's called very low risk or low risk disease, and whom we think that it may never affect them. And that's really one of the main issues that creates the controversy around prostate cancer screening, is to identify those cases that need to be treated are what's called clinically significant prostate cancer, as opposed to those lower risk cases that may never affect a patient in their lifetime. So, although there are many markers, and many of you may be familiar with, what's the ROC curves that look at biomarkers that basically show the sensitivity compared to the 1- specificity. For those who don't know, a useless marker is a sort of diagonal line right up the middle. A perfect marker would go up, straight up and straight over. And as you can see, that many of these biomarkers that are newly developed, and there's really an explosion in biomarkers to try to fill this void and this need, that these really are not perfect, and I would say many with similar imperfect results. What about imaging? So MRI has what's called multiparametric MRI of the prostate. Has become a mechanism or a way that we initially evaluate a patient very commonly now in a man who has suspicion for prostate cancer based on his PSA level. And that's because the MRI can help to identify areas of suspicion within the prostate. This is the MRI. This is the MR spectroscopy, which is not commonly used, but was in this paper certainly. And this was a paper from the NIH where a lot of the research, initial research on MRI and the pathologic outcomes were done. And although MRI is very helpful, it's not also is not perfect. And what we can see is that there are false positives where not every lesion or imaging finding is absolutely prostate cancer, and there can be false negatives in which there are significant cases of prostate cancer that we just cannot see, that are invisible by imaging. And how do we use the information? Practically speaking, for our patients, if we see that there's a finding on MRI, we actually can. This is an image of what's called a fusion biopsy. It's actually, this is probably in terms of diagnostics for prostate cancer, it's been a real advance for patients and for we urologists who take care of patients, where there's software, and there's now a few software platforms that we can use the MRI, which is done in advance of the biopsy. That MRI is prepared, the images are prepared, and then when we do a biopsy, we do it under the guidance of an ultrasound. And then at that time, we use the software to overlay the MRI findings with the ultrasound findings, and we can target those areas of suspicion. So we know that MRI targeted biopsy and use of this pathway has helped us reduce the, I would say, the negative biopsy rate, and has helped us to avoid unnecessary biopsies in many men, and has also improved our detection rate of clinically significant prostate cancer in men. So it certainly has helped, but it's not perfect. And this is a paper that we had written looking at comparison of fusion biopsies, what I was talking about, where you use the MRI information at the time of the biopsy and comparing that to the final surgical pathology after the prostate's out and you see the whole thing under the microscope. An important thing to know is that the biopsy is just a sampling, and there can be sampling error, and you don't always have the exact full picture as you would imagine when you take needle biopsies, it doesn't always represent exactly the entirety of the story that's going on within the prostate. This, not to go through in detail just because of being short on time a little bit, but just to say that even those targeted biopsies don't always give you the entire picture of what's going on with the prostate cancer, and you can sometimes miss areas of higher grade disease. Sometimes you can overcall the severity of the disease as well. So it's not absolutely perfect. And again, this is just further information to highlight the potential, for false positives on imaging and false negatives. So some of you may, if you're at all interested in prostate cancer and keeping up with literature and even sort of lay press in prostate cancer, you may have heard about PSMA imaging. PSMA is prostate-specific membrane antigen. It's a targeted imaging using PET scanning. There's both using PET CT for looking for distant or metastatic disease, and then the question if can we make our regional and local staging of using MRI better by combining PET, PSMA-based PET with MRI. And likely we can make the characterization a little bit better. But from a screening standpoint and from an evaluation standpoint of patients who don't yet have a diagnosis of prostate cancer and at risk, we always have to think about what's really the cost of all of these advanced imaging techniques, and how we deal with that from a public health standpoint, I think is important to consider. So urine-based biomarker discovery and analysis is something that I've had a long interest in, as I think Bruce knows and Andreas know, that I've through fellowship years ago and spending time in a mass spec lab, Steve Giggy's mass spec lab at Harvard Medical School, and Bruce Zetter's lab, and doing proteomic research and some metabolomic research also, but really have spent a lot of time through the funding from the PCF and DoD trying to identify novel biomarkers. And the reason urine is I think an attractive platform and I guess medium to identify biomarkers. It's non-invasive. It's a simpler proteome, or I would say even pan-ome, even metabolome than serum, and it's a filtrated blood. So the kidneys, which produce the urine, receive 25% or a quarter of the cardiac output or blood flow, and so really that filtrate carries many either proteins, metabolites, volatile organic compounds, which really can then serve as an area for biomarker discovery. In addition, as I think, as Bruce pointed out, the urethra or urinary tract goes right through the prostate. So there will be contributions from the, in what we call a first void specimen, directly from the prostate into the urinary tract. And similarly for kidney cancer and bladder cancer, this is an area where biomarker discovery may be very helpful. This is just when I first met Andreas, I was at the PCF maybe now, I think a few years back. I was enthralled by his and amazed by his talk because in 2004 I actually had shown this slide to my department when I was when I was a senior resident and doing proteomic research and biomarkers, and this fascinated me at that time, and the ability to now participate in some of this research now so many years later is really a thrill for me. So we know, just to summarize, PSA-based screening and detection is the current standard. We know it's it's very imperfect, and there's a huge void that I think needs to be filled by other, in addition to sort of thinking, classic biomarkers, algorithms that we can use to detect disease. We've demonstrated the importance of early detection, as I talked about in the recent non-screening era and non-PSA era, which I think are coming out of, hopefully. And we do still struggle with imaging and who to use imaging in. We want to make a cost-effective approach and really, be mindful of limiting cost in populations of men, and we really need to identify more accurate, non-invasive, and affordable diagnostic strategies, really, that I think are critical in assessing men who may be at risk for prostate cancer. So again, thank you to Andreas. I'll stop sharing.
Thank you, Adam. That was very thorough, and it dovetails so well with a previous presentation on Crohn's disease also. I think much to unpack here. Please stay for the Q&A at the end of the hour. So for the next talk, it is my very strange and distinct pleasure to introduce Dr. Simmie Foster. who also happens to be my wife and mother to our three wonderful children. And she's right here next to me. And I'll put her up in just a second. So you can just say hi. I asked Matt to rename my screen from Dr. Merchant to Dr. Simmie Foster. And her talk today will be on olfaction and neuroimmunity. I know I embarrass her every time I say this, but she's one of the propagators and perhaps establishers of this field of neuroimmunity, which is the language that the nervous system talks to the immune system. And it's very important to recognize that these two are actually connected. And the olfactory apparatus is part of the nervous system. So Simmie will talk about olfaction and neuroimmunity, scent as an adjuvant in vaccines and something that to do with mental health. So I welcome her here, and I will share, please come in, and I will share your screen for you. Excuse the clunkiness here, as we share one home and one computer. So here's us sharing our screen in just a second. Yes. If you
Correct.
All right, so Simmie, you can drive it by pushing these buttons right here, and I'll fade into the background. Take it away.
Thank you so much, Andreas. I definitely did not invent the field of neuroimmunity, but I do study it. And I'm very interested in my lab looking at how the nervous system and the immune system really talk to each other and how they coordinate immune responses, and especially how sensing of the environment can modulate these immune responses and change how our body responds to things like infection, and how it changes our moods. So I'll talk a little bit about how all that relates. And the first thing I'm going to talk about is vaccines, which recently have been a bit controversial, but we all probably know a lot more about vaccines than we used to. One thing you might have heard of is that the first sort of modern vaccination was done by Edward Jenner in the late 18th century. I think it was like 1796, according to this picture. And since there were no rules about clinical research back then, what he'd observed is that smallpox was a huge problem, and it killed a lot of people and also disfigured a lot of people. So most people had smallpox scars. And he observed that milkmaids actually had this beautiful skin with no scars, like very few scars. And it's because they would catch cowpox. And then they wouldn't catch smallpox, and cowpox was a much milder disease. So he wondered if he could take lesions from someone who had cowpox and vaccinate, in this case, the young son of his gardener, who I think was like 9 years old at the time and probably didn't want to have stuff stuck in his arm, but he was probably coerced. So he took some cowpox from one of the milkmaids and injected into this young kid, and then later on exposed the child to smallpox, and the child did not catch smallpox. And so he started to do this very widely, and it became a practice of vaccination. And the principle is that if you inject something that is not going to kill you or make you too sick, then your immune system can generalize and then make a response to get rid of the big baddies. So I will go to the next slide.
Is it a problem?
Yeah, I'm just like, there. Andreas has a new fancy computer. So I told, as I mentioned, in my lab, we're studying the neuroimmune system. And it used to be thought that vaccination was mainly a product, like the vaccination response was mainly a product of the immune system. But what we're finding is that neurons and immune cells really communicate extensively in every aspect of the immune response. And so it really is a neuroimmune response. And you can find neurons in close proximity to immune cells in every tissue of the body, from like, for example, T cells can regulate blood pressure in the heart, and there's macrophages in macrophages are these innate immune cells that are in every tissue of the body that really help maintain homeostasis in concert with neurons. And not only do sensory neurons and immune cells monitor homeostasis, so general health of tissues, they also help defend against pathogens. And as Andreas mentioned,
Next slide.
No, I'm good.
Sorry.
As Andreas mentioned, the olfactory bulb is part of the brain, right? So our ability to sense scents is a neuronal capability that also is involved in immune responses. And this is just beginning to be explored. But you can imagine that like the olfaction might coordinate behavioral immune responses. So for example, you can sort of smell if someone smells like sick and sweaty, just like we've been talking about the whole conference, and you might stay away from them, but also directly coordinates immune responses. And many immune cells have olfactory receptors. There's also these specialized epithelial cells that have taste receptors and olfactory receptors, and both maintain homeostasis of neurons and also actively coordinate immune responses. And those are called tuft cells. And they're found in many epithelial surfaces, including the gut, the respiratory epithelium, and some places in the brain. So what does this mean in terms of developing vaccines? So one idea is that we could use scents as adjuvants. So what are adjuvants? Adjuvants are known as the immunologist's dirty little secret. And this was proposed by Dr. Charlie Janeway, who is my mentor's mentor. And an adjuvant is basically something that tells the immune system that a certain substance is dangerous. Because if you just take, like, for example, a COVID spike protein, like there's nothing about it that says, oh, I'm going to get you. I'm dangerous. So you need something else that goes along with it. And in an actual viral infection or a bacterial infection, that can be things like tissue damage. It can be things like particles from microbes that just aren't present in our normal eukaryotic cells. For example, in bacterial cell wall, or LPS, is not present normally in healthy tissues. And so the body can recognize that and say, oh, this is a harmful and virulent pathogen that the immune system needs to respond to. So an adjuvant is basically something that tells the immune system that it needs to make an actual defensive response. And just as an aside, I was wondering what the COVID-19 vaccine, mRNA vaccine adjuvants were. And it turns out that many of the companies are using nanoparticles, which might have precipitated some of the controversy about tiny little chips, because nanoparticles sound a little scary. And it's a little bit unclear exactly how they're acting as an adjuvant, but one theory is that they cause intracellular damage. So they damage lysosomes, which then there's sensors inside your cell, such as the inflammasome, that then are turned on by this. And then they make cytokines, which then activates the immune response, and then coordinated with the production of the spike protein says, okay, this spike protein is dangerous. So for olfaction, we can ask, can signals from actual olfactory bulb, olfactory sensory neurons, or from extraneuronal olfactory receptors that are present on other cells like immune cells, tuft cells, other places, can we make cocktails of these or activation patterns that then serve as an adjuvant? Can we design scent combinations that can make vaccines better? And just in support of this idea, there is a nice story in Drosophila that in Drosophila, there's a odorants, and it's unclear which odorants, but generally, like the ability to smell actually is essential for the development of these cells called lamellocytes that protect against wasp infection. So if you get rid of the ability to smell in Drosophila, they can be infected very badly by parasitic wasps. And there's these odorants basically stimulate neurotransmitters that then coordinate the development of the lamellocytes. Okay, so this also could have implications for not just vaccines, but for inflammation and for mood in general, and for pain. So how might that work? So, in an immune response, or in a neuroimmune response, you might have an immune stimulus such as bacteria or a virus or whatever, and it can talk to cells such as macrophages that then produce cytokines such as IL-1 beta. And IL-1 beta in particular has a lot of actions on the brain. and many other places in the body, and like instigates inflammatory responses, all causes fever and sickness behavior and pain. So the sickness behavior is basically people don't want to go out. They feel really crappy. They like their mood is down. So they really look a lot like a depressed person. And the IL-1 beta itself actually sensitizes nociceptors or pain neurons and leads to pain. So all that achy feeling when you have an infection is really because of these little molecules called cytokines. And there's some indication that that you could probably modulate production of things like IL-1 beta and the inflammasome using using odorants, using olfactory stimuli. And that might be the basis behind some of the effects of aromatherapy. For example, eucalyptus oil has been used for thousands of years to loosen up mucus and make people feel better when they have congestion. And it also can be directly antiviral, and as well as probably activating antiviral immune cells. And so in this case, we're sort of making an anti-adjuvant, which is calming down the neuroimmune system and leading both to better mood and to could possibly lead to reductions in pain. So these are just some of the things we're looking at in my lab for hot and cool research. I think olfaction is going to be a very interesting avenue into learning how to control our neuroimmunity and finding better therapies and probably making better vaccines. So I'd like to thank everyone. Thank Andreas very much for inviting me. And it's been wonderful to speak to you today.
Thank you, Simmie. Let's swap seats for a second, and I'll stop sharing our screen here. What a weird thing to be to have this arranged. Let's see. Oh, it was stopped the whole time. Oh, maybe somebody else stopped it. Okay, so now we're going to open it up for questions, and I can have you maybe come here because some of the questions will be addressed to you.
One more speaker, Andreas.
I'm sorry. Oh, we have one more speaker. I'm so sorry. Of course. Oh, very sorry. Cecilia Roque, and I'm so glad to have her here. She is going to talk to us about a technology that I was not familiar with. I believe your crystal sensing for olfaction is just. She has created a fantastic video, and she is the associate professor. And excuse the problem with the pronunciation here, but if I could say Universidade Nova de Lisboa, is that correct? Okay, then. So please introduce yourself properly with a proper accent, and share your screen, and I will unpin myself. And Matt, please pin Cecilia. Thank you. Go ahead, take it away. You have 5 minutes or a little bit more.
Okay, so good afternoon, everyone. Here in Lisbon, it's already afternoon. Your accent was almost perfect, Andreas. So first of all, I really want to thank Andreas and the Osmocosm team, because I think the organization of this meeting is going really very well. And I think what is quite interesting from what I've seen is that, you really managed to grasp the different dimensions of olfaction. And I think it's really fantastic to have the opportunity, to listen to all these different perspectives, and even now, just this talk that we've heard, the relationship with immunity. So it's really fantastic. So my name is Cecilia Roque. I come from the Nova University of Lisbon, and we have been also working in the field of artificial olfaction for a while, and we are actually trying to convert the unseen into something that you can see. So that's why I gave this title to my talk. So trying to see the unseen with the technology that we are developing. So, I come from Lisbon, I don't know if you ever visited, but if you haven't, please try to do it. It's a place where you really have the opportunity to have quite a lot of fragrances and others around that, make your experience quite unique. And we also have, lots of nice beaches around. And so it's a nice place for you to really wake up all your senses. And what I think that is really beautiful, and let's say it's also what makes it so interesting to study about olfaction, is that we are studying things that we can't see. And I mean, obviously humans are very visual. So, I mean, there are, even the sayings that if you don't see something, you can't believe in it. So that's why probably, olfaction, it's one of these senses that it's more difficult to study and to understand, and to actually, be able to give these little triangles and squares, and to understand that these are really chemical compounds, and what's this composition of this mixture, and what's the fingerprint of this mixture. I think it's really what we want to try to understand. So what is actually our vision in sensing? So we are using basically this sentence that a nose that can see is worth two that sniff. And so this is why we are trying to have a nose that can actually see. And to do so, I mean, we have been working basically with materials that are quite unconventional. So we use something that everyone likes, I believe, gelatin. And we use also a specific type of solvents to dissolve this gelatin. And we combine this with liquid crystals. That, I mean, we know they are widely used, for example, in liquid crystal displays, etc. And so the combination of these three components actually gave rise to a mixture that actually has what we call the cooperative assembly, meaning that you only have the assembly into micron nanostructures in these materials when we have all these components together. And this also allows molecular recognition, in particular VOC molecular recognition. And so the materials that we work with and that we use as gas sensors are basically gel-like materials, as you can see here. And if you look inside, so we have like the polymer matrix that creates these long chains, and we encapsulate in this gel these droplets, these purple droplets, and these purple droplets are actually liquid crystal molecules encapsulated. Yeah, and they have as obviously an interface, in this case in the ionic liquid that is making this interface. And the liquid crystal here works as the oil phase that basically gets entrapped inside this little droplet. And we can really see, if we just slice the gel and we just look it, under the microscope, we can see these little droplets. They look like, chocolate chips in a biscuit. And when we look under polarized optical microscopy, we can see that these nice little, droplets of liquid crystal, have this nice appearance, and they correspond to these little mountains, in the gel. And when we look, in more detail, we can really see that we have these, very beautiful images. But, this is really nice and cool to see. But what is the relationship between, these nice optical textures that we have and VOC recognition and sensing? So basically what we play with is with order and disorder inside these type of materials. And this order and disorder is basically given by the presence of the volatile organic compounds, and they create a certain dynamic function to these materials that can be used for sensing. So for example, in this video that I'm playing, we can see one of these areas under the microscope, and what we've seen was basically the exposure to a certain volatile. I'm going to play it again. So we start with, the beautiful, this beautiful image of the liquid crystals. We then basically apply a certain VOC, and we can see it basically becomes completely black, and then it comes again to the original position. So this is the basis of what we do in this technology. So this is under a polarized optical microscope. So it's already very beautiful images and you can play with them. We went a bit further. So what we've done was basically to use these materials as sensors, both electrical or optical sensors, that we can, we build an entire device where we can, use these little sensors and expose them to volatile organic compounds mixtures. And the interesting thing is that the way these nice liquid crystal droplets change their optical textures is not the same for different compounds. So if we just study in detail, for example, hexane and diethyl ether, which are different volatile organic compounds, we can see that the way this particular liquid crystal droplet changes the textures, the optical textures upon exposure and then upon recovery, which is the second step, we see that it's different from diethyl ether, and we can pick up these differences in the optical signals that we actually collect from the device. And so we can either play around with the signals that we get from the device itself, and obviously build classification models that allow you to distinguish with high accuracy these different compounds. Or we can look not at these signals that we can collect from the device, but we can also look just to the optical texture of a single droplet that we see under the microscope. And we were quite surprised that, just looking at the droplets, at single droplets, we can actually get quite a lot of information just making image analysis. So in this particular case, we used some sequences for classification and others for test, of course, and we got, a really nice accuracy in the classification and also in the prediction of concentration of a particular compound. So basically what I've shown you is the technology we were working with in the field of artificial olfaction. It's a very unconventional way, let's say, to look at it. And so I really want to thank, obviously, the really nice team that I have working on this. And thank you very much for your attention as well. So looking forward for, questions.
Professor Roque, thank you so much, Roque. Your team, I think, wins the award for most beautiful Osmo data visualization. Okay, absolutely, hands down. I am embarrassed to compare your data to how my data looks, which is all a bunch of nerdy stuff. That is definitely the future. I am so excited that you gave this. I would also ask you to put the link to your very nice video that you have created, which goes on for 6 minutes. Please put it in the chat for people to watch later. Now, unless Carolina tells me otherwise, I think we're done with the speakers for this section, and we are going to invite them all back for the Q&A. And we have so many questions for so many people, and I'm going to try and have at least one question for each person because there's so many that have been coming in. And I'll start with the first speaker, Bruce, who gave a talk on the evolution and how we're trying to outrun cancer. And Bruce, I have a sort of I don't know if it's a philosophical question or what, but you showed a cancer from 2 million years ago, evidence of that. We've been battling this thing for 2 million years. Is there something wrong about us thinking about it in the way that we do? Is there a way out? Are we ever going to get rid of this horrible thing that is affecting humanity? And if so, what do you think is the future of that struggle?
No, I don't think we're going to get rid of it. I think, we're going to get better at finding it when we can easily cure it, the way Adam was talking about, and what this whole sensing technology is focused on. I think we will develop better treatments that can extend life and quality of life for people with cancer. But this is something that's part of us, essentially. It's in our genes. I was so focused on trying to stay within 5 minutes that I sort of forgot to say my main point. And that really is that our bodies developed these metabolic programs over millions of years during periods where it was hard to get enough nutrients. You had to expend a lot of energy. and you didn't live very long, you know. And one of the things that we have now is our metabolic pathways have to cope with the fact that they were still thinking about, you know, living on roots and grubs. And we're talking about protein shakes and McDonald's. And we're living until 70, 80, 90, when our immune system stops working as well, our nutrient absorption stops working as well. And evolutionarily, we're nowhere near, you know, adapting to all of that. So I think all of these things are part of what contributes to cancer, you know, forgetting even about environmental pollution or contaminants in food or preservatives. It's with us, and, you know, until our lifestyle starts to, until evolution catches up with our lifestyle, it's going to be a problem.
Well, thank you for that. And it's a little bit pessimistic, I think. Here, let me offer a more optimistic, perhaps, take. Stumbling and falling over when you hit your foot on a rock is also evolutionary forever with us, okay? And we've, even Lucy, our last universal common ancestor that we found bones of, she fell into a big cave in Africa, in South Africa. And so stumbling as you walk is also preventable in the sense that, okay, if you break your foot now, we can fix it. So my next question goes to Adam. If you were to fantasize about a future where you don't have to do as much perhaps damage or as many invasive procedures to your patients, what would it look like? Do you feel that we can maybe cure prostate cancer and other cancers and other urological disorders by detecting them early? How does that look? And can we get closer to, okay, we can't get rid of cancer completely the same way that we're always going to be able to fall over and if we stumble on a rock. However, we no longer die from an infection. We no longer die from a scratch. Can we see a future where doctors like you are empowered by some technology? Because what is evolution is great, but what we're forgetting is that we have evolved technologies too, and those might be able to help us. So Adam, what do you think about that?
So great question. So I actually want to follow up, and I was waiting to follow up on what Bruce was saying, because I completely agree with Bruce. Unfortunately, I do. And the reason is, similar to viruses, malignancy and the developmental malignancy and the biology behind that is incredibly heterogeneous. And there are so many different biologic mechanisms that are at play when a cell makes that transformation to becoming malignant. And not only that, but, why some patients, develop metastases and some don't, and why some develop metastases at 3 years and some develop metastases 20 years later, why that occurs. And, likely our immune system is, in play in regard to sort of clearing, out those cells. But so I agree with Bruce that it's not going to be, I don't think we're ever going to completely cure all cancer. I unfortunately don't, but I think we're going to become much better at detecting it early and managing it. So the question is, how do we do that? I think immunotherapy is a very exciting advance recently. I think that the ability to utilize our body's own immune system to fight cancer, I think, is going to be an important and continue to be an evolving area for us. One of the potential, every treatment has a potential side effect, and how we maximize that and minimize those potential sort of autoimmune side effects that and inflammatory side effects that occur from immunotherapy, I think are going to be important. If we think about how do we get away from me having to make an incision and cut out the tumor, for lack of a better term, there's an evolving area of focal therapy and minimally invasive therapies, but those are also really dependent on good and accurate imaging. And so I think that's important, where we can use targeted imaging. And that's what I was getting to with some of the PSMA imaging-based modalities. But so I think, there's been a huge advance in the past several years, and I think there's a lot more to do, because just the heterogeneity, there's so much we don't understand yet.
I'm so glad.
Andreas, let me inject a note of optimism.
Yeah.
And it comes from elephants. One of the reasons that we haven't evolved to escape cancer is because most cancers occur way past reproductive age. So there isn't a natural selection against cancer. But there's something called Peto's paradox, and it's named after Richard Peto from Oxford. And the idea is that we think that the more cells you have in a body, the more likely you are to develop cancer. Bigger mammals develop cancer more than smaller mammals. Why don't elephants develop cancer? So it turns out that in humans, there's one gene that has a major role in taking care of damage to DNA, and that's the PTEN gene. And there's one of them. Elephants have 12 different PTEN genes. So they can get rid of a whole lot more DNA damage than we can. So, maybe over time, just as development of metabolic programs required expansion of the genome, maybe, through some miracle of who knows what, we're going to do the same.
Some miracle of technology is what you want to say, I think. Thank you for that. That is very hopeful, in fact. And as soon as you said it, I was thinking of the technologies that are coming out of my lab and also Kate Adamala's lab in UMN, where she's talking about gene copy manipulation and the 3D printing genes into vessels that are what are called SINELs, synthetic cells. Now, my next question is going to go to Simmie. Because Adam set it up for me so perfectly. He talked about immunotherapy, and I want to ask Simmie about neuroimmunotherapy. Please, can you slide yourself over and tell us what you think about neuroimmunotherapy? Can we, and as just to pile on you because you're right here, I would also like to ask your thoughts on nerve involvement and neuro stimulation as part and parcel of cancer, right? We've seen that people who are experiencing different temperatures, which is Simmie's lab is called the Lab for Hot and Cool Research, because she's looking at the effects of heat transfer and temperature variations from everywhere from single cells all the way to whole humans. So where is neuroimmunotherapy in this, and where is sensing and comfort and cancer together?
Oh, wonderful and very broad question.
Yeah.
So first of all, one of my colleagues has a very nice paper on how nociceptors and immune cells together actually detect cancers. And many cancers, we had this observation actually that oftentimes a painful lymph node, for example, means infection, while a painless large lymph node can mean like metastatic infiltration. So we started wondering whether there's something to do with the nociceptors that somehow the cancer cells are interacting with nociceptors. And what my colleague has found is there's this interesting interplay between checkpoint receptors on immune cells and nociceptors and tumor cells. And I believe, I'm going to try not to get this wrong, but I believe that oftentimes the tumor cells are actually silencing the nociceptors and sort of like telling the nociceptors, I'm not really here. So the nociceptors can't recruit in the immune cells that would generally be detecting the tumor cells. So that's one point. So yeah, I think there is probably co-surveillance of that neurons are doing as well of tumors. The second point is hyperthermia. So like heating people up has been used as a therapy for various tumors for a long time, because it was observed that many tumor cells have a different heat tolerance than the other cells in your body. And so you could use targeted hyperthermia to shrink bulk tumor a lot of the time. And what's interesting is that there's a little bit discussed about what role the immune system might play in this, but not much. And hyperthermia in general has varying effects on different cells in the immune system. One of the things that we think might be playing a role is that immune cells and other cells in your body can sort of protect themselves from high temperatures, like they induce these heat shock proteins that sort of hold everything together, while tumor cells, that whole system is a little dysregulated. And the tumor cells that are resistant to hyperthermia are able to upregulate heat shock proteins. This also has implications for other things such as Alzheimer's, where heat shock proteins might actually help the protein, like the native protein, stay in its native conformation rather than generating plaques and tangles. But that's another discussion, not specifically related to cancer. And on the other hand, there's not a lot studied about cryotherapy that I've been able to find. Most of the research has been on heat therapy. And one of the things that I'm really interested in is how neurons might be, there's trip channels on neurons that are sensitive to heat. And it turns out that it's these temperature sensitive neurons that seem to have a really intimate connection with recruiting and activating immune cells. And so what role are immune cells playing? What role are these temperature sensitive neurons playing in both like the therapies for tumors and for the immune response in general?
What a wonderful and broad response there. And sorry for setting you up with such a broad question. My last question is for Professor Roque. And again, what a beautiful readout system you have. And actually, I have so many questions. I want you to maybe take this offline at some point with me. And I would like to collaborate with you in many ways to create better noses. So one question I have is, how do you stabilize these crystals? And can we deploy artificial intelligence, such as what Stephen Thaler is doing, to look at them and start maybe deciphering not only what cancer, et cetera, but even perhaps on the mechanism of why it is that they're changing in response to those volatiles the way that they do. And where do you see your technology going?
Yeah. Thank you, Andres. So, basically regarding the use of image to analyze, these nice videos and that we can get. It's obvious it's something that we are doing. So I've just shown these recent data that we got in collaboration with a colleague that is really expert in deep learning analysis in images. And we were really surprised what we can just learn, just analyzing, single droplets, and how this can actually give you a very high accuracy in not only in the classification, but also then in the quantification. Then in terms of, from a mechanistic point of view, trying to understand at the molecular level what is going on. This is something that we are quite curious as well. So we have started doing some tests with, molecular dynamic analysis and coarse grain. So to try to understand exactly what is going on. And it's a very complex system, but it's also very cool to study. But it's definitely ongoing, because there's so many more things that we can actually do with the system that we've built. And where do I see it going? We are obviously interested, and that's we are also doing that at the moment. Doing a lot of proof of concept tests to see how this behaves in much more complex other environments. So we've done it, for example, for monitoring the food getting rotten, and it's so it means you can really smell things going on.
Absolutely, yeah. And I want to also point out that I think that your visualization of the data, it's beyond just cute and beautiful. It is actually closer to what I imagine the nose is doing in the olfactory bulb and the epithelium. It is really the activation of these 400 different types of neurons in these patterns that look very much like your like your crystals. I am surprised and delighted by this technology. And so we've reached the end of our official Q&A session. So what happens next is we have a 40-minute lunch break. So people should eat and power their brains for the next session. We will have the Zoom running for with some background and people can chat, and we're still collecting questions from everyone, and we will pose them to the speakers as necessary. And if they want to jump in and answer, go ahead, use the chat. At 1 o'clock, we will welcome Dr. Shuwang Zhang from MIT, who is also the head of the Laboratory for Molecular Architecture. And then we'll hear from Ilya Kolb, who's Dr. Kolb, who's the senior project manager at the Romix, which is a company that creates pallets of cells that express olfactory receptors for flavor, and they're moving now into medicine, possibly. We will listen from Dr. Wenhee Lee, who is at the University of Texas at El Paso, and she is with myself and Adam and Bruce and Stephen Thaler and Constantinos, Papa Constantino and Howard Sewell. I hope I'm not forgetting anyone, in the company called Sentient, which is here, by the way, .ai, which is an unfunded startup, which has not yet been, it's sort of in stealth mode up until this Osmocosm conference, and we hope to solve a lot of the problems associated with early detection of prostate cancer. We'll also listen to Lucas Lopez from Volatile AI, which have created a handheld, I would say this big machine that can do chemical analysis via scent. Dr. Joseph Azzarelli will talk about distributed olfaction for augmented awareness, and he's been around this conference the entire time. We'll thank him for that. And finally, we'll talk about on-demand bioproduction of aromas to enhance flavors and food experience for astronauts. A problem that you didn't think you had, but we do. So with this, I want to thank our panelists and speakers of this morning. You've been great. Thank you so much for contributing to Osmocosm. We hope to see you again next year, and we hope that you remain engaged in this community. And we congratulate every one of you for your successes and fantastic speeches. Thank you. Have a wonderful lunch. We'll see you at 1 o'clock, which is in 37 minutes.